Hello, everyone. It is 2:02 p.m. in the afternoon, and this is the MPRINT webinar series. Welcome, everybody. Today, we will be talking about biologic medications and autoimmune disease. We have Dr. Anna Patrick here, from an assistant professor of pediatrics and rheumatology at Vanderbilt University Medical Center. Dr. Patrick completed her MD and PhD in molecular biophysics at University of Texas Southwestern Medical Center, followed by her pediatric rheumatology fellowship at Vanderbilt University Medical Center. She recently became funded through a NIH NIAMS K08 award and has received funding from the Arthritis National Research Foundation and the Childhood Arthritis and Rheumatology Research Alliance, which helped her establish a basic science research program to study the pathogenesis of pediatric autoimmune diseases. Dr. Patrick's research focus is on identifying and characterizing pathogenic molecular pathways in pediatric autoimmune disease and in juvenile idiopathic arthritis specifically. Her research program employs the study of rare monogenetic mutations associated with disease manifestations to discover novel pathways involved in autoimmunity. This strategy defines the relationship of these pathways to the broader disease population. In her juvenile idiopathic arthritis research, she also works to understand the relationships between biologic phenotypes and therapeutic responses, with a goal to personalize medical care for children with arthritis. She is the recipient of pilot study funding through MPRINT to better understand the genetic contributions to transcription of inflammatory pathways in juvenile idiopathic arthritis. In this talk today, Dr. Patrick will be discussing the interesting characteristics of biologic medications as a category and how this relates to their use from a pharmacology standpoint. Dr. Patrick will also talk about the advent of biologic medications, their use in treatment disease, and the challenges associated with the rapid development of new therapeutics. Dr. Patrick will address special challenges with biologic medications in the pediatric population at the end. Dr. Patrick, welcome, and go ahead and get started.
Hello, can you all hear me?
Yes, we can hear you.
Awesome. Let me. I am unable to share my screen or turn on my video.
You're muted, Lindsay.
Go ahead and try now.
Okay. That is solved. Okay, great. So hello, thank you for that kind introduction. So my name's Anna Patrick, and I am a physician scientist in the Division of Pediatric Rheumatology at Vanderbilt University Medical Center. As was mentioned, I've been supported by MPRINT as part of the Precision Therapeutics Academy and through support for my projects. So I'm going to talk to you about something that impacts every day of my clinical life and most other days. And the topic is biologic medications in autoimmune disease. So first, I have no conflict of interest. So the goals for the talk is we're going to talk about biologic medications. And as I was doing this, I realized that we're really going to be focusing on antibodies as biologic medications. We'll talk a little bit about autoimmune disease and then the intersection of biologic medications and autoimmune disease. And then we'll talk about the benefits and challenges of biologic medications. And we're going to use juvenile idiopathic arthritis as an example. So my first thought when I decided to present on this topic was, how much are people talking about this, right? So what I like to do whenever I think about what is the interest level in a topic is just to go to PubMed, type in the words and see how many citations appear over time. And so I just type this in for biologic medication, antibody, and autoimmune disease. And while you can see that there are little specklings back to like the 1900, there are very few. It's very flat up until around the 1950s. You start seeing a little bit of an uptrend in antibody, a little bit of an uptrend in autoimmune disease, and then you see a giant spike upwards for biologic medications. And so this is something where the interest is rising, the topic is rising. It's clearly, I mean, these are almost vertical lines for biologic medications. And I just went back to see, well, what was the very first one of these what is the very first publication that showed up with these? In biologic medication, it was mentioned in 1894. And I'm not sure if this is really biologic medications as we think about it, but it's a very interesting kind of discussion about skin problems. And antibodies were mentioned in 1901, and you can see that the English is completely different than how we would use it today. But again, a very interesting thing. And then autoimmune disease first showed up in 1942. So one of the ways to think about this is first to ask ourselves, what does the Food and Drug Administration actually consider to be a biologic product? So what are biologic products? These are vaccines, blood and blood components, allergenics, somatic cells, gene therapies, tissues, and recombinant therapeutic proteins, which would include antibodies. These products are made of sugars, proteins, nucleic acids, or complex combinations. It can also include cells and tissues. These are isolated from a variety of natural sources, or they can also be produced using biotechnology means, and that becomes important. Often these are at the forefront of biomedical research. So how does a biological product differ from a conventional drug? So in contrast to most drugs that are they're chemically synthesized and their structure is known, biologics are not like that. They are complex mixtures. They're not easily identified or easily characterized. A lot of biologic products also tend to be heat sensitive, and they're susceptible to microbial contamination. So you may have heard about biologics and biosimilars. This becomes really important. And when we think about the FDA, we need to think about what are the definitions for these things. So a biologic product is going to be the reference product. It's a single biological product that's approved by the FDA. And this is the product that a biosimilar would be compared to. Importantly, in a biologic product, there are lot-to-lot differences. And so these need to be carefully monitored. Then if you think about a biosimilar, they are a biologic product very, very similar to the reference product with no clinically meaningful differences. And but it's important to remember biosimilars can have other slight differences in things like the clinically inactive components, which can be really important for reactions to the products or things like antigenicity. So, and then there are things called interchangeable products. An interchangeable product is something it's a biosimilar that has met the additional requirements, and it can be substituted for the reference product without the prescriber being a part of that substitution. So what's important is biosimilars, they're not just generic biologics though, okay? They're still a very complex drug. So I'm going to end up focusing on antibodies, and IgG-based products are really how most antibody drugs have come about. And so if we look at regulatory approval, this timeline is going back to around 1975. And at first I'm just highlighting, this is on the top is this is when monoclonal antibodies were first produced using hybridoma technology. And this was done by Milstein and Kohler, and this ended up being awarded a Nobel Prize. This technology really, really started this entire thing. And then we learned about antibody chimerization was first described. And then followed by that, we learned about how you can humanize antibodies. Phage display technology occurred in like the 90s-ish. And then there was descriptions of transgenic mice with human immunoglobulin genes. So in this very beginning of this, and this is not that long ago, just between like 1975 and the 1990s, this is when the technology really ramped up for these products to become drugs. And all of the things after that, most of these are just noting specific drugs and when they occurred. And I put stars next to the drugs that we commonly use in the rheumatology clinic. So it's very clear we use these drugs heavily. So in order to really talk about antibodies as drugs, first we need to talk about an antibody. So an antibody is part of the host cell's defense. It's made by a white blood cell called a B cell. The structure of the antibody consists of 2 light chains and 2 heavy chains. And at the very tip of them is the hypervariable region. The hypervariable region is the part that binds to an antigen. So it's important to know antigens can be things that are foreign to the body. It could be a virus, it could be a bacteria, and in some cases, your body could actually appear as foreign for an antibody. And so that might be something called an autoantibody. So the other thing that's really, really important to remember is antibodies are really heavy. This is so much bigger than what we think about as a chemically synthesized drug. Antibodies are around 150 kilodaltons, and they are globular plasma proteins. So for all the different types of antibodies, the basic structure is the same with the heavy chains and the light chains. These chains are connected by disulfide bonds. For the light chains, they're around 22 kilodaltons, and the heavy chains are around 50 kilodaltons or more. There are 5 types of Ig heavy chains in the mammal, and these are denoted by the Greek letters, the alpha, delta, epsilon, gamma, and mu. And then there are 2 types of light chains, and these are lambda and kappa. So when we talk about isotypes for antibodies, which you can see on the right-hand side of the screen, there are 5 types. We've got IgG, IgM, IgA, IgD, and IgE. These are classified according to the heavy chain constant region. And they're distributed throughout the body and they function differently. Importantly, if you look at, is the pointer, can you see this laser pointer?
Yes, I can see it.
Okay, perfect, thank you. So this variable region is, you'll often see it written as FAB, that's the antigen binding fragment. And this constant region you'll see written as FC, which is the crystallizable fragment. And you'll see it denoted that way later in the talk. So other things that are really important. So when we look at this, the IgG is where most of our biologics are going to be based off of kind of this structure. There are 2 antigen binding sites, and these are the 150 kilodaltons. This is 80% of your total antibody in your serum, okay? It can cross the placenta, it can fix complement. The FC region, that constant region, often binds phagocytes. This is our main blood antibody. It's involved in secondary responses. It can neutralize toxins. It can be involved in opsonization. So this is important when we think about how can we leverage an antibody, which is a natural part of all of us, as a therapeutic. So there are different formats for antibodies as a biologic medication. The first one is the canonical antibody. This, and most of the antibodies that have been approved fall in this category. The second one is an antibody drug conjugate. You can imagine how important this might be in something like cancer, where you want to bind a certain cell type and then, noted by the X and crossbones, have something bad happen to the bad cell. You can have a bispecific. Remember that normally our antibodies are the same on both ends, but a bispecific might bring 2 things together that are different. And then you can have fragments of an antibody. And so these fragments become important when you think about it changes the binding specificity, how you have this set up. It can change the size of the drug, and that's going to be really important for how it works in the body. And there's only 4 listed here, but there are a tremendous number of antibody formats that are not listed here. For example, we don't have radio-labeled antibodies here. We don't have antibody immunotoxins shown. And there's at least 30 more bispecific formats. There's a lot of bispecific formats. So I thought this was, the first thing I wanted to talk about was this very interesting paper. This just looked at the FDA's first 100 antibody approvals. And it gives you a sense of how have antibodies, just how have these drugs occurred, and what are the kind of historical patterns for what's happened. So the first one was approved in 1986, and it was a canonical antibody. And these are just looking at using just those 4 types we just talked about, plus an other category. And you can see that most are the blue color, which is canonical antibodies. You can also see that this is definitely rising over time, right? And over time, we're now seeing the appearance of antibody drug conjugates, bispecifics, and fragments. So these antibody biologics are being developed. So what is the therapeutic area for which they have an approval? Vast majority are in cancer. These drugs are really big in cancer. And there are some that are dermatology, hematology. What's important to see here is that these antibodies as drugs, they traverse many different therapeutic areas. Cancer by far is the biggest. However, for example, in rheumatology, many of the drugs that were approved for cancer trickled to rheumatology. And I'd like to point out that if you look at this slide, this is basically a slide for adult medicine. These are pretty much never approved for pediatrics first. This is always approved for adults first. Okay, so what are the targets in these 100 antibodies? Because this kind of helps us understand what is the thought process behind how these are being developed. So the top 10 targets for the first 100 monoclonal antibodies encompass 42% of all of those antibodies. So these are not 100 unique targets. These are highly overlapping targets. You can see PD-1, PD-L1, which we've probably all heard of. These are mostly or almost all used in cancer. CD20, which is going to be a B-cell target used in cancer, also used in lots of rheumatology indications. And then going down from there, lots of rheumatology indications and other things that are easily associated with cancer. So our targets are overlapping. So what does that mean for the development of new antibodies? Are we developing a bunch of new antibody targets, or are we developing things against all the same molecules? What I think was really interesting is if you looked at the top investigational monoclonal antibody targets, they were pretty overlapping with the ones that already had good drugs. So PD-1, PD-L1 had 80 investigational agents, and probably this paper was from a couple of years ago, so probably some of these are now approved. But at least 4 of them showed up on the top list of investigational antigen count or agent counts. And so this tells you that we have some really good targets that seem really effective, and companies are jumping on those targets and modifying the antibodies and changing the antibodies and seeing if they can do things just a little bit differently to make a new drug. So why are we doing this? Why are companies so interested in these monoclonal antibody targets? And it's because these are huge money, right? So in this third table, this is looking at the top monoclonal antibodies by 2019 sales. And you can see what the antibody, the target, and then these are billions of dollars in 2019. And the first one's a TNF, the next 2 are PD-1 targets. I mean, this is huge money. And I put a star next to all of the ones that we prescribe in rheumatology. So why are these antibody therapies so attractive? So we have the specificity and the affinity for the secreted and cell surface targets is pretty amazing. There are all these different formats available. And importantly, these are more likely to succeed in trials than small molecules. So the phase 1 to approval success rates ranged from 17 to 25%, depending on the therapeutic areas. And so if you look over here on the right, this is looking at the monoclonal antibodies that entered clinical study between 2005 and 2014. There were 569 of them. The green looks at all, and then these, along the y-axis is percentage, and then green is all, purple is non-cancer, and red is cancer. And the first one looks at phase 1 to 2, phase 2 to 3, phase 3 to the regulatory review, and then regulatory review to approval. And then the very final one looks at the phase 1 to approval, like how many drugs actually made it from beginning all the way to end. It's really amazing that around 22% of them made it. So that's about 1 in 4 actually made it all the way to approval. And if you compare that to new molecular entities, that's at best a 1 in 8. And so these monoclonals are being granted marketing approvals at around twice the rate of small molecules. And that really, really makes it financially beneficial for companies to pursue these as drugs. So it's important to understand the antibody mechanisms of action so that we can understand how they work as a drug. So they can neutralize, you can take advantage of the FC-mediated effector functions, you can take advantage of immune complexes that trigger downstream humoral and cellular responses. So in the first part, in A, we're looking at neutralization. Here you have an antigen, this little triangle. It's coded by immunoglobulins, which are antibodies, and then the pathogenic interactions of that antigen with your body are inhibited. In B, we are looking at antibody-dependent cell-mediated cytotoxicity. So when this antibody binds to an antigen-positive cell, it has an effector cell that interacts through the FC receptor, and then this results in killing of the target cell. And then in C, this is complement-dependent cytotoxicity. So here you've got activation of the complement component C1q, and this triggers a complement-dependent cytotoxicity and the killing of this cell. So these are just some of the mechanisms that can be leveraged in order to either normally to eliminate pathogens or disease cells like infected or cancerous cells, but these actions can be taken advantage of by designing antibodies for specific disease processes. So when we consider an antibody as a therapy, what kind of things do we need to think about? So first, we need to think about our choice of target. Then we need to think about what is the desired antibody effector function that we are aiming for. We need to think about what is the antibody affinity. So it's important to remember affinity is the strength of the antibody-antigen interaction. And we need to think about the binding avidity. And this is the total combined strength of the interactions between an antibody and its target through multiple binding sites. Remember, IgG had 2 binding sites, but other IgG types like IgM, they have far more. And so that's going to change the way that the antibody as a drug interacts with the antigen and how long it might stay at that place. You need to think about the product size. This becomes incredibly important when you think about clearance of a drug and things like that. So it also is really important for tissue penetration. And then you need to think about what kind of interactions are you wanting to take advantage of through the crystallizable fragment. There are normal body interactions with antibodies that you can take advantage of. So one of the things that we think about when we talk about drugs are the half-lives of our drugs. So antibodies are very complicated because of their size. And so when we think about an antibody half-life, what are the things that become important? So one of the important aspects of IgG is that it can bind to the neonatal FC receptor, and you can kind of see a little depiction of this on the right. So the neonatal FC receptor is on endothelial cells in the liver, also on some other cells, and it uses endosomal recycling of the antibody product, and this results in an increased half-life in circulation. So you can see how you have the IgG, a protein it's bound to, it goes into the endosome. In the acidified endosome, our IgG gets sorted, the proteins get degraded by the lysosome, but then the IgG gets recycled out of the endosome and goes back into circulation. So this can dramatically increase your half-life. So you can imagine how modification of an IgG to change its interactions with the neonatal FC receptor could be used to either increase or decrease the half-life. If you think about avidity, then you can think about how the residence time of an antibody at a surface is going to potentially result in it being longer in the body. It can also slow, tissues are difficult for an antibody to penetrate through because of their size. But if you have this binding, it may have longer residence time at a tissue. And then if you think about other ways to extend the half-life, you can think about adding things to an antibody so that you can have steric hindrance to reduce proteolysis and to make the product size greater than, for example, some type of renal filtration threshold. These are pegylated proteins. You could add bulky polypeptides. You could conjugate to fatty acids. And there's really a lot of ways that this can be done. So this is the IgG subclass, and these are the cellular Fc gamma receptors. So you can see that there is IgG 1, 2, 3, and 4. And I'm not going to go through all the details of how these have small differences. The important thing is that these different IgGs bind to these different Fc gamma receptors. Some of them, there's a high affinity receptor. Monomeric IgGs bind to these differently than immune complex IgGs. So you've got monomeric receptors. You also have one that is, these are low affinity, and then you have one that is inhibitory. Then you have one that's got no intrinsic signaling. So these receptors are very different, and by selecting an antibody with different properties or different subclasses, then you're going to be able to potentially shift the interaction of that antibody with the normal cellular physiology that could have different interactions with that drug. So how can we take advantage of this? I think that this really highlights antibodies as a biologic drug that is really complicated, but that has so many ways that we can modify the antibody to make a drug that does what we want. So on the left-hand side, this is looking at ways that you can design antibodies to try and improve homogeneity and the potential for their development. So there have been a lot of studies looking at antibodies and how we can improve their performance, both in the production, processing at all the different stages. So this has resulted in identifying kind of minor antibody components, for example, charge variants, glycoforms, disulfide bridge isoforms, and other aspects that can help to improve the stability or the pharmacokinetic and pharmacodynamic properties of an antibody. And this is how you can make the next generation of an optimized antibody. You might be able to get greater homogeneity, stability, or potency. And so each of these little areas is kind of highlighting one of those types of things. For example, if you change the hinge region, you may get some better properties for your antibody. So that's just talking about homogeneity and potential for development. So then we can also think about how we can improve the pharmacologic functions. And when we think about that, you can use the structure-function relationships to allow fine-tuning of these properties. You can think about how you could tailor this variable domain so that you get better or lesser binding to your antigen of choice. You can also do things to try to increase or decrease the half-life of your antibody. You could also choose to only have an FAB so that you do not have the larger macromolecule. There is a lot you can do. And then, of course, conjugation of things to the FC region, conjugating radioactive nucleotides, chemotherapies, toxins. There's really a lot of different ways that people are actively modifying antibodies. And so this kind of answers the question of, well, we have these good targets, and then we look at where are the new targets being developed, and many of them are being developed to the same kind of antigen. So you say, well, why are we all focusing on PD-1 and PD-L1? Why are we continuing to make new antibodies? It's because there is a lot that can be done to an antibody to change the way it functions, depending on what you are looking for in your for the diseases you're treating. So this brings us to the next part, and this is to talk about antibodies and autoimmune disease. So an autoimmune disease happens when the body's natural defense system can't tell the difference between your own cells and foreign cells, and that causes the body to mistakenly attack your normal cells. So I just wanted to list a multitude of the autoimmune conditions, and probably people have heard of most, if not all of these. And as you look at this, you notice that it crosses all different areas of the body, and some of them are systemic and involve multiple areas. And if you're familiar with autoimmune disease and you look at this just very small subset I've listed up here, you'll notice that there's a whole lot more that are more rare, less common. I've left out a tremendous number of diseases. So if you take all of the different autoimmune conditions and you add them together, about 1 in 15 people in the US have an autoimmune disease, and that's a huge number of people. Importantly, and we're not going to focus on this today, but we know that many autoimmune diseases are more common in women than men. So when we look at that other slide, it's really focused on a lot of those are more common in adults. And then if we look at pediatrics, okay, so autoimmune diseases, they can definitely happen in kids. So we have some that are more organ specific, and we have some that are not as organ specific and involve multiple systems in the body. Today, what we're going to end up using as an example is juvenile idiopathic arthritis. And a lot of these over here fall into the category of a rheumatic disease. And we'll talk about that later. So autoimmune diseases are very frequently the targets for biologic therapies. So why are biologics so useful in these diseases? So first, the immune system is critical for autoimmune disease. Many of the immune cells are in circulation. And then immune cells, cytokines, and all the different signals that they use, they are all targets for antibodies that you could leverage as a drug. And then you can take advantage of the antibody functions after you identify your targets. And additionally, many of the autoimmune diseases, they don't have good, effective alternative therapies. So there's not just an easier way to treat this disease. And many of them just flat out haven't had good therapies at all. So what is a rheumatic disease? Rheumatic disease, it's an umbrella term. It refers to arthritis and other conditions that tend to affect the joints, tendons, muscle, ligaments, bones, and muscles. Many of these diseases also affect multiple organ systems. So what are our common rheumatic diseases? In the adult clinic, the most common is rheumatoid arthritis, and in the pediatric clinic, the most common is juvenile idiopathic arthritis. So there's a shared feature here. These are autoimmune arthritis, and so autoimmune arthritis occurs across the age span. So what are some of the drug targets in autoimmune arthritis? These arthritises come from environmental triggers and genetic factors. We have different immune cells that are involved and different local and systemic effects downstream of these. You'll notice that we have the things that I've circled are some of the biologics that are important at targeting some of these different immune cells. So in autoimmune arthritis, we have a tremendous number of biologics that are major drugs to target these diseases. So now I want to talk about juvenile idiopathic arthritis and use it as an example for talking about antibodies as a biologic. So juvenile idiopathic arthritis, which I'm going to call JIA, it's our abbreviation, it really is a group of inflammatory disorders. The shared feature is a chronic arthritis of unknown cause. Symptoms begin before age 16 and persist for longer than 6 weeks. and it can't be caused by something else. It can't be caused by infection or cancer or some other thing. So I don't know if you guys have ever seen a kid with JIA. So these are two pictures. This little child has oligoarticular JIA. They have arthritis in one big knee. And this child has polyarticular JIA. That means lots of joints involved. And so you can see the wrist, most of the joints in the picture are involved. In this picture is showing jaw asymmetry. So your TMJ can also be involved. So when do kids get diagnosed with JIA? On the right-hand side of the screen, I'm showing patient number versus year of life. Purple is girls and blue is boys. And you'll notice there's a peak incidence of diagnosis in the prepubescent time frame before the 6 years of life. So between 1 and 5 is our most common age for diagnoses. And interestingly, we see more girls diagnosed than boys. And this is prepubertal. So we don't understand this, but I think also, again, something for us to make sure to always take note of and pay attention to. So JIA is not one thing. It's clinically heterogeneous. And we're not going to focus in on this slide a lot, and we're not going to get into the nitty-gritty of how to diagnose this disease. But what I want you to take away from this is that there are different subtypes of JIA. And these subtypes, at the bottom is kind of a Venn diagram showing the overlapping features between subtypes. Some subtypes overlap a lot, and some subtypes don't overlap a lot. Some of them overlap more with some of the adult arthritis diagnoses, and some overlap less. So this is really important when you start thinking about drugs to treat a disease. Some categories of JIA respond to similar medicines, and some categories respond to different medicines. So this is just showing the disease-modifying anti-rheumatic drugs and then the biologics for JIA treatment that were available in 2015. The ones in blue at the bottom are all things that really we don't recommend anymore. One example I like to point out is that gold was the original thing to treat arthritis, but we really don't do that anymore. And then we look at the blue are approved for pediatric arthritis, and then the lighter green tells you which subtype or which disease it's approved for. You'll notice these subtypes are not universally spread across as far as FDA approval. And then the pinks are approved for adult arthritis, but we use them in kids. And then the blue are approved for adult arthritis. And now this is 2015. So this is kind of old news. Some of these are now approved in kids and we're using them. So why are biologic therapies so attractive for JIA? Number one, it improves disease outcomes. These drugs work. They treat previously untreatable disease. They can have an additive effect on traditional DMARDs like methotrexate. They are overall really well tolerated, and they help us as a steroid-sparing therapy. In pediatrics, chronic steroids are really bad for growth and development. So if you have a 1-year-old diagnosed with polyarticular JIA, you have to treat the disease or they're going to have joint deformities and other problems for the remainder of their life. And if you treat them with steroids, they're going to have negative impacts on growth and development. So using our DMARDs and using our biologics has really been a game-changing therapeutic advance in our field. And that's really highlighted by this figure. In this figure, it's showing the results of pivotal phase 2 and 3 trials. These were trials conducted by the Pediatric Rheumatology International Trials Organization and the Pediatric Rheumatology Collaborative Study Group. And these were in collaboration with pharmaceutical companies. And in the first, importantly, when you look at these trials, these are all trials for antibody biologics, right? And then if you look at this, the red is subcutaneous, the blue is IV. All of these are injection drugs. You cannot take an antibody orally. Your stomach will just degrade it. So these are all IV or subcutaneous. And you can see how many of the patients in these trials responded with an improvement in their JIA. And then you can look on the right, and this shows you the placebo versus drug. Placebo is in yellow. These are patients who had a flare of disease during the trial. So you see more flare under placebo than under these biologic medicines. And these two images just highlight how effective and important the development of antibody biologic drugs has been. So there are a lot of challenges when you want to use a biologic medication in children. So first, none of these drugs were developed for pediatric autoimmune disease, just none of them. You have issues with demonstrating safety and efficacy. These are in large part rare diseases. While JI is the most common disease in my clinic, it's still relatively uncommon in the general population. So this makes clinical trials very difficult. In addition, we have these heterogeneous disease populations, which makes clinical trials even more difficult. Then sometimes you run into the ethics of the placebo arm. If you have such an effective therapeutic, is it ethical to give a child a placebo when it could negatively impact them to not be treated with the drug? So you have to think about that in clinical trial design. And then we think about dosing challenges. Is a 1, 2, 3-year-old going to need the same or a different dose than an adult? And it's important to recognize that there have been examples where you need the same dose. There are doses that you need to have a lower dose. And there are sometimes for certain medicines where you need a higher dose. So an incredible advance for us were the Best Pharmaceuticals for Children Act, the BPCA. So what did this do? This incentivized pharmaceutical companies that voluntarily perform pediatric studies by providing the opportunity to gain 6 extra months of pediatric market exclusivity. So this tried to stimulate work for kids, but by using money, basically. And then we have the Pediatric Research Equity Act. So this was mandatory. It authorized the FDA to require pediatric assessments for certain drugs and biologic products unless the assessment was deferred or waived. And so these two acts were very, very important for pediatric drugs. And then even more important is that the FDA Safety and Innovation Act made these permanent. So they were kind of recommendations, but then they got made permanent. And so that kind of solidifies the ongoing success of these acts. So since 2012, these two acts have resulted in more than 900 products with specific pediatric use information in their labels. So I'm going to go back to challenges. I'm showing a few challenges and then a little bit of data and information about the challenges. So for this, we're going to talk about dosage forms. These, the antibody biologics are shots or infusions. And then you have to think about adherence to your medications. These are all specialty medications. Some of them have pretty complex administration. So when you think about barriers to medication adherence, what I'm showing on this screen is parental endorsed barriers. So the parents recognize these barriers, and then patient endorsed barriers. So in this study, they collected data from 578 parents and 99 patients. Some of them, 44 were parent-child dyads. And when they collected this data, about 70% of patients and 70% of parents all recognized at least one adherence barrier. And what I wanted to point out about this is there's lots of different barriers, but it really stands out that for injections, pain was recognized both as a parent and a patient barrier. It's interesting because these injections, most of them subcutaneous, have this as a barrier. But if you look at infusions, which are IV infusions, either at home or in the hospital, they really don't have that barrier. And another barrier that was the black is oral meds, white is injections, and hatch marked is infusions. If you look at worrying about future consequences, patients worried about future consequences for any of the medicines pretty equally. Parents worried about injections a little bit more, but it also was kind of a higher amount of barriers endorsed as opposed to some of the other things. So when we think about antibody therapies, we're thinking about injections and infusions, and pain and worrying about the future are some of our major barriers. So additional challenges in biologics. The cost is prohibitive. It is very expensive. All of these drugs are expensive. And what that means is that you need to have insurance coverage for these drugs. So this also means you end up needing a lot of prior authorizations for drugs. Insurance companies will require another medication or another treatment failure before you can get the drug that you really want to use. That becomes a problem because we know that as arthritis continues, the severity of your arthritis, the longer your arthritis is not under control, the harder it can be to get it under control. So having delays in treatment is really bad. So the other thing is sometimes insurance companies have changing coverage for biologics. They'll cover it 1 year and then they'll switch to something else the next year, or they'll switch to the biosimilar. And we really have to be conscious about switching back and forth between a bunch of different medications. And whenever you see cost of a medication being so high and complexity of a medication being so high, we really have to think about health inequities. And then we have a paucity of data for length of treatment. We know when to start the drugs, but we don't really know when to stop them. So if you're 2 years old and you start a biologic medication, we don't have great data on when you should stop that medicine or if you should stop that medicine. So this is a study that looked at the economic impact of JIA. So they found 18 eligible studies with around 6,500 patients. So what they did is they took and I'm only showing you a subset of the paper right now. So they took your original currency and then they converted everything into US dollars, and they cost adjusted based upon year. And in this table 3, this is just showing the total costs, and it's looking at a breakdown of the different categories of JIA. And I put a star next to polyarthritis. You can see that polyarthritis in multiple groups was more of a total yearly cost than the other categories. And then if you look to the right, this breaks it down a little bit differently, and these are different studies. If you looked at without biologic therapy versus with a biologic therapy on average was more expensive than not having a biologic therapy. And most of our biologic therapies are antibodies. In particular, at the time this study was done, that would have mostly been antibodies. And in this study, this looked at childhood arthritis and cumulative social disadvantage. And there's a lot to unpackage about this study. But what I really liked about it was that this is from the a cross-sectional analysis of the National Survey of Children's Health. So that is a less biased study analysis. Rather than just going and looking at patients with arthritis, they looked at kids. So they looked at around 130,000 surveys, and 365 of the kids reported current arthritis, kids or their parents. And then they took the kids who had arthritis and they looked at cumulative social disadvantage. And the components of that were a low household income, low guardian education, underinsured status, and a high adverse event in childhood score. And on the left-hand side, we're looking at the individual components, looking at the odds for arthritis diagnoses. And you'll notice there were individual components that having that component gave you a higher odds ratio for an arthritis diagnosis. And then if you look below it is the individual component score with the odds for arthritis severity. And what you'll notice is there were individual components that made it more likely for you to have a higher arthritis severity. So this is really important information. And so then they took these scores that they generated by these 4 things and called that the cumulative social disadvantage. And so you would get either a 0 if you had none of those things, or a 4 if you had all of those things. And then they did unadjusted odds ratios or adjusted odd ratios. And the adjusted odd ratios used a multivariate model adjusting for age, sex, race, and ethnicity. Importantly, you get the same result in both cases. And what you see is that the higher your cumulative social disadvantage was, you had a significant increase in your odds ratio for an arthritis diagnosis. Or the same thing, the higher your cumulative social disadvantage, you had an increased odds ratio for having moderate to severe arthritis. And this is really important because it doesn't tell us what came first or what came second. But this starts to give us an understanding of arthritis and how your social disadvantage might be contributing or playing a role in your arthritis. And when we think about biologic medications and the cost and complexity associated with those medications, and we start combining that with social disadvantage, we have to really think about this and think about it in the terms of how do we make sure to diagnose and treat patients to the best of our ability. So I'm going to end with, it's not really a high note, it's mostly a problem. So this is a challenge in biologics and JIA, that is anti-drug antibodies. There are also other challenges. For example, it's unclear which patients will benefit from which biologic. Sometimes biologics stop working, but we're going to focus in on the anti-drug antibodies because sometimes that's why a biologic stops working. So we have these complicated antibody biologic medicines that we're using. We give them to people by intramuscular or by subcutaneous or infusions. And sometimes people's body develops your own antibodies against that drug. So on the right, this is showing different antibodies and the identification of anti-drug antibodies. So first, they've been identified against multiple biologics. This is not a problem that has been solved. It can impact the efficacy of the biologic. We really don't have good clinical or really even research measures to easily identify anti-drug antibodies. And so that brings up some really big questions. Should we monitor them? There are a couple of clinical tests for them. If someone was doing really well on a drug, and then they start developing new arthritis, and we test for anti-drug antibodies, and they have new anti-drug antibodies, it's likely that that's one of the reasons for why their biologic is failing. So that makes you think, okay, well, maybe anti-drug antibodies are helpful. And then there are other cases where you measure anti-drug antibodies in a patient who's doing well, and they have some anti-drug antibodies. So it's really complicated data to know exactly what to do with. And that also brings up this last point. Should we treat through? Should we increase the dose? Should we stop the medication? While we do have multiple biologic medications to choose from, we do not have an infinite supply of biologic medications to choose from. And so if you begin developing anti-drug antibodies to medicines when you're 2 years old, by the time you're 20, you may have run through most of the medicines that are available. So that's just kind of food for thought. So summarizing what we've talked about, we talked about biologic medications with a heavy focus on antibodies. We talked about autoimmune disease and a little bit about pediatric autoimmune disease. And then we illustrated the benefits and challenges of biologic medications in pediatric autoimmune arthritis. And with that, I've put my full references are each included on the bottom of the individual slides within the red stripe. All right. Does anyone have any questions?
That's exactly it. Thank you, Dr. Patrick. Does anyone have any questions? I actually have one for you. As an infectious disease doctor, I've seen 2 cases now of PJP in children with rheumatological disorder and on a biologic, not on glucocorticoids. And so there's always some question, should we be prophylaxing these patients? Has there been any push in your realm to start standardizing prophylaxis and with the increased amount of biologics on the market?
Yeah, so first I'll tell you, I'm not sure that everyone does anything the same way. So maybe that if you ask 10 people, you get 10 slightly different answers. The way that I think about this, and I think, so first, this is an excellent question. In particular with PJP, this is a drug that there is a preventive measure. You can give people Bactrim. There are things to do, right? So we have some options for preventing. So that's the first thing, is we have options. The second thing is, in my kids with rheumatologic diagnoses who have lung involvement, a lot of them end up on prophylaxis. When you are giving someone heavy steroids, Cytoxan, you're bottoming out their immune system. Those kids are also pretty easy to make a choice on, that they need immunoprophylaxis, or they need prophylaxis for PJP. And I tend to almost follow some of the HIV/AIDS guidelines and look at CD4 cells, because those are really well-established guidelines. So I kind of like to look at those. The other thing is thinking about Rituxan. If you take away all of someone's B cells, tend to prophylax those guys. And then if you have a vasculitis, sometimes they get prophylaxed. So you kind of select, you pick and choose. When I think about juvenile idiopathic arthritis specifically, there are children with systemic JIA who have systemic involvement, including severe lung disease. Those kids get prophylaxis. And I have seen that specific disease as an example where we saw some infectious things creeping up that are very hard to diagnose because they look inflamed, which can come from their rheumatic disease and from their infection. And when they get an infection, their rheumatic disease becomes more active. So you have just a circle of inflammation that you're dealing with. I know that wasn't a perfect answer. There is a great recognition for a need for better standardization, but without really good studies to help us know who is going to benefit the most or who is going to have a side effect to the preventive medicine, I think you're probably going to find people doing slightly different things a lot.
Thank you so much. That answers my question perfectly. Does anybody else have any other questions? I don't see anything in the Q&A or chat. So if nobody has any questions, thank you, Dr. Patrick, for joining us today. We really appreciate it. This is being recorded, so it will be available in the near future. And if there are any questions that pop up afterwards, please email us at Megan Creaser, and then we can direct them to Dr. Patrick. Thank you so much.